of oxidative phosphorylation. Let's go to this last slide we were on. Okay, so I think this is where we ended. We didn't get into the last chemiosmosis step. So as a reminder, this final step called oxidative phosphorylation sort of has two little substeps within it. The first being the electron transport chain. And the electron transport chain is exactly what it sounds like it is. It's a chain where electrons are transported along it. And as a reminder, when these electrons are transported along this electron transport chain, that's going to release energy. So electrons being transported just through those proteins are releasing energy. That's really important because that energy is pumping hydrogen ions, these H+, across this inner membrane of the mitochondria. So we're in the mitochondria, we're in that innermost membrane where all of this is happening. So now on one side of that membrane, you now have a lot of hydrogen ions or H+ ions. And that'll come into play in the final step that I'll go over on the next slide. We're just about done with all those electrons that we shuttled over with the NADH and the FADH2. So they get passed down this electron transport chain. They end up on oxygen. So oxygen is that final electron acceptor. So they get passed along here, end up on a molecule of oxygen. That basically results in the production of water. So oxygen will bind with a couple of hydrogens, and you get water as a product. Okay, so now we've kind of followed the path of the electrons. I've talked about the hydrogen ions being pumped to the intermembrane space, to one side of the membrane. So now just imagine we have all of these hydrogen ions up here, just a huge concentration of hydrogen ions on one side of the membrane. And if you think back to basic diffusion, those hydrogen ions are going to want to come back across the membrane. They just naturally want to diffuse back to where there's fewer of them. So that's what our next step is. Wait a minute. Yes.
Where are the hydrogen ions being pushed to?
Oh, they are a good question. They're in the mitochondrial matrix, which means like the most central part of the mitochondria. They're being pushed to the intermembrane space. So in between the two membranes of the mitochondria. If you imagine the inner and the outer, right in between those. Yeah, good question. All right, so now let's look at those hydrogen ions and see what they're going to do. They are, like I said, going to diffuse back across the membrane that they were just pumped across, essentially. They're doing that not just through any normal kind of protein. There's a really specialized protein that will be used to allow those hydrogen ions to come back in across the membrane. And it's called ATP synthase. It's this crazy looking rotating protein. It's really complicated, but you just need to know the name of it. and know that that's where, so remember in that inner membrane space, in between the inner and the outer membranes of the mitochondria, there's all these hydrogen ions. They want to come back across the membrane just to spread out and have a little bit more room. So they're just going to flow back down through ATP synthase, through simple diffusion. So for you guys on Zoom, up here we have this high concentration of hydrogen ions in the intermembrane space. They're going to diffuse back through this protein, ATP synthase. And as you might imagine, based on the name, ATP synthase is where we're actually going to synthesize a lot of ATP. So you can think of ATP synthase sort of as a motor that's powered by these hydrogen ions. So when a certain number of hydrogen ions come through it, I'm not going to go into the details about how it all works, but hydrogen ions are coming through it. It makes the rotor spin, essentially. So like I said, it's like this little kind of mill within the membrane. So it actually makes it spin. It's powered by the hydrogen ions diffusing back through. And that's what actually creates ATP. So without getting into too much detail, when that motor spins, that basically allows for this extra phosphate to be added to one of our ADPs, which are just circulating around, that adenosine diphosphate. To make it a triphosphate, obviously we want one more phosphate group. Instead of di, we want tri. So by that motor, the ATP synthase basically rotating, that will allow for ADP to combine with another phosphate group to make ATP. I'm skipping over a lot there, but that's, I think, the most important part. Knowing that hydrogen ions are flowing through, making this motor basically spin, that allows for these two, ADP and P, to come together, and you get ATP. That's the process in a nutshell. So for you guys on Zoom, if you can see, I was pointing to this ADP here. Adenosine diphosphate is combining with this phosphate that's hanging out on its own, and that's going to form your ATP. And that is the process of chemiosmosis. Sorry, I didn't define that. So chemiosmosis is this process of the hydrogen ions flowing back through the membrane. And that is the last step of respiration. If we look at one adult person, this is a rough estimate, depends on obviously what size this adult person is, but 133 pounds of ATP are basically recycled this way every single day. So about the weight of many adult people. The ATP basically being formed and then being used again for other processes. So we recycle going from ATP to ADP, using it and making it, about 133 pounds of that per day, which is crazy. It's a tiny little molecule. So that being said, we do it a lot. We make a lot of ATP and we break a lot of ATP down. Okay. I'm about to talk about, oh, well, yeah, let's look at it. I'm going to zoom out a little bit here and kind of look at the whole process of, this will be sort of all of oxidative phosphorylation. So those two steps where you have the electron transport chain, and then you have your ATP synthase chemiosmosis. So just to review, this last step of oxidative phosphorylation, we start out with the electron transport chain. Electrons are brought in on NADH. Remember, that's our electron shuttle. So NADH here is bringing the electrons that we need. along with FADH2, sorry. So those two electron shuttles are bringing over electrons. The electrons are flowing through this electron transport chain. And these little bursts here are just basically illustrating the energy is released. I kind of like that part of the diagram. So you have energy being released when these electrons are passed along this chain. That energy is being used to shuttle hydrogen ions out into the space between the inner and the outer membrane of the mitochondria. So here you can see we're in the intermembrane space up here, the mitochondrial matrix down here. All right, so now we have all of these hydrogen ions out in the intermembrane space. They want to come back in. So they're going to diffuse back through ATP synthase. That's our chemiosmosis step. That's going to rotate that motor and then allow ADP and a phosphate group to come together, and we make ATP. Okay. Any questions about that? We're so close to the end. So close. All right, so let's kind of summarize what we made. Well, this is it basically. So for our purposes, we're saying 32 ATP are produced by the oxidative phosphorylation, so ETC plus chemiosmosis. We make 32 ATP and 4 waters. The waters are just the result of, like I said, oxygen being that final electron acceptor. So oxygen will grab onto electrons, that combines with some of those hydrogen ions, and you get water as a byproduct. So the outputs of this last step are super simple. A, you get most of the ATP. That's the take-home message. You get the vast majority of ATP from this oxidative phosphorylation step. And then water. So that's it. That's all you get from that last part of ATP or of respiration. So now let's look at the whole chart all put together, all the steps of respiration when we're starting with 1 molecule of glucose. So this is basically just combining all of those other, weird, gray's not showing up. This is just combining all of those other steps that we already went through, basically all the other tables that I've already put together. So we've talked about this first part enough, I think. So glycolysis, pyruvate oxidation, citric acid cycle. That gives us, so what I have here is substrate level. That means substrate level phosphorylation. So if you look back at the free energy lecture that we went over, I talked about two different ways to make ATP. There was substrate level phosphorylation and oxidative phosphorylation. Substrate level was a lot less efficient. Oxidative phosphorylation is a lot more efficient, meaning you get a lot more ATP. So for the total substrate level phosphorylation, we made 4 in those first few steps. Remember, pyruvate oxidation is just an intermediate. I put it on its own line, but it's not technically one of the 3 main steps of respiration. It's kind of to get from glycolysis to the citric acid cycle. And then here I just mentioned the electron transport chain. So that's electron transport chain plus chemiosmosis. I'll update this. I don't like having these differences in my terminology. So I'll update this and upload that version to Canvas. So we have 32 made in that last big step of respiration. So that gives us a total of 36. We have a couple places where we made CO2, so don't forget to take note of that. So kind of it's made back to back. Pyruvate oxidation and then citric acid cycle. Then we also have water coming out. So now we've accounted for all of these molecules, right? So we came in with one glucose, one C6H12O6. We need 6 oxygens, which I didn't really talk about. That's just at the very end where oxygen is that final electron acceptor. That's why we need 6 oxygens. We make 6 CO2, 4 and 2. So 4 in the citric acid cycle, 2 in pyruvate oxidation. So we've got that covered. 6 waters, 6 H2Os. We've got 2 in glycolysis and 4 in the last step, oxidative phosphorylation, basically. So 2 waters up here in the first step and then 4 in the very last. So that gives us our 6 waters. And then the 36 ATP, which we calculated in the ATP column. The NADH and the FADH2 just kind of keep getting recycled. So once they release their electrons in that electron transport chain, they've shuttled them over. They basically just go back to sort of the beginning where at the first step they're needed and do the same thing just all over again. So they shuttle, release their electrons, go back, pick up more, shuttle them over, release them, etc. So that's why they're not down here in this reaction. All right, any questions about these numbers? Anything you're wondering where the numbers came from? Totally confused by. It's a lot, I know it's a lot to look at. Okay. So this is just the one slide, basically, that I'm going to talk about the fact that we do eat stuff other than glucose and we break it down as well, right? I mentioned this at the beginning when I said we're really going to focus on glucose, taking that through respiration. When you eat fats and proteins, they also get broken down through cellular respiration. They just kind of come in at different points. You don't need to know what points they come in at. I just wanted to take a second so you're not like, oh, all we eat is sugar and we only break down glucose. I want you guys to realize that yes, fats, fatty acids, or glycerols, they'll basically kind of bypass that glycolysis step because they don't need to be broken down like glucose does. So they'll come in sort of later in this whole process. Proteins, same thing. So they'll come in just a little bit later. I just think that's an important point to at least mention. You don't need to know details about when they come in or what they turn into. Just know that yes, we break down fats and we break down proteins as well, all through cellular respiration. They just skip some steps. They come in a little bit later, but we can make ATP if we're breaking down fats and proteins as well. Okay. Let's see. Okay. So this is I think the last slide I have on here. I know this looks like a lot. It's basically a cartoon. I don't know where it came from. There's these weird ant things. I don't know what they are. I got it from someone a long time ago. But it breaks down each of the steps. So if you can sort of follow through in far less words than I just spent explaining this, right? It breaks down the main steps of what's happening in glycolysis, kind of shows some images or some drawings of what's being produced at each step. So this summarizes it. There's a lot going on, but there's a lot going on in respiration, so it's kind of hard to simplify it too much. So if this is helpful, this is sort of the super condensed version showing you what you start with, what's made at the end of each step, where CO2s are made, where ATPs are made, and how many of each are made. So like I've said, I think I've said this in here before, I'm really visual, so it helps me to, if I see something like this, I can remember, okay, there's 4 CO2s coming off the Krebs cycle. It's easier for me to remember this picture than it is to remember a bullet point that says 4 CO2s. So if this is helpful for you, awesome. If not, you can ignore it. And then we're going to watch the Amoeba Sisters summary of respiration. So they do a really good job of kind of walking you through respiration. This will be a review of kind of the whole process. So hopefully this is a nice wrap up to the breakdown of kind of how I did it. If you don't remember some of the stuff they're talking about, that's perfectly fine, but hopefully this will kind of get your, you'll remember as you're watching it and get your brain kind of on track with the big picture, kind of zooming out, because in class, I sort of have to zoom into all of the little details. So this will kind of zoom out and give you hopefully a bigger picture view of it. I think that was the last slide. Yeah, okay. So we're gonna watch that right now.